Thanks, Dr. Tsai. We appreciate that. That was really, really very nice. From the very beginning, one of the implications as far as the gene drive systems are concerned was the possibility of using them under environmental conditions, and therefore what the implications of that would be. We have two individuals that are going to talk to us about that now in terms of understanding ecological and evolutionary conditions for gene flow. And we're going to start out first with plants. Dr. Allison Snow from Ohio State University, who has made a number of important contributions in this area. So Allison's going to get us started now. Okay.
Thank you. I'm really glad to be here to talk to the committee and the broader public about these issues. I chose this slide of a dark rain cloud over one of my research sites because that's kind of how I view the technology right now. I think it's very powerful and awesome, but I don't understand it well enough to know that it will be safe to use, and that's kind of a theme of my presentation. I'd like to start with some background about genetically engineered crops, since that's where we have the most expertise with releasing GEOs in the environment. And just remind you that it's been fairly recently that we've had these crops available, and starting in, well, I call it recent, 1996. You can see from the adoption rates here. But we don't have very many crops that we're familiar with. It's mostly corn, soybean, canola, cotton, and a few others. And not very many traits have been put into these crops, mainly herbicide resistance and insect resistance genes, and some disease resistance genes. So in terms of trying to understand what these transgenes are going to do out in the environment, one of the questions that my research group and a lot of my colleagues has worked on is gene flow. And not very many of these crops have wild relatives, so we don't really have a lot of experience in tracking transgenes, but I'll tell you what we do know. And some of the examples that are already out there are canola and squash. The slide on the right is just a photograph of what a lot of Ohio looks like, and a lot of the Midwest. It's dominated by Roundup Ready soybean and corn, and here you can see no-till agriculture in action, where last year's corn has not been tilled, and it's possible to plant the soybean right on top of that and spray it with Roundup and kill all the other weeds. So in terms of our gene flow research, the same questions have come up around the world. Anytime you have a genetically engineered crop and wild relatives present, people want to know where are those genes going to go and what impact might they have if they get into wild or weedy relatives of the crop plants. Here's a schematic just summarizing a lot of work that we've done over the years, where we know that if the wild or weedy relative is closely enough related to the crop plant, pollen can carry the transgene into existing weed populations. This is an example with sunflower. With sunflower also, the crop itself can persist in seeds that come up, and we call those volunteers. Sometimes those volunteers can persist and start new populations, which might be called feral populations. And so gene flow can take place via all of these different routes and also back from the wild ones into the crop. So we've seen all of these examples using different kinds of molecular markers in different systems. And basically, transgenes will move in the same way. Most of the transgenes that have been introduced into crops don't have a fitness cost per se when they get into a wild organism. And they might even help the wild organism if they confer something like insect resistance or disease resistance or herbicide resistance. And we've seen all of those kinds of examples. So we've been studying that. At the same time, I'm an ecologist, and in 2005, I worked with the Ecological Society of America to come up with a position paper on looking at genetically engineered organisms very broadly. So this was 10 years ago. We had an interdisciplinary team of co-authors, including Jim Teege, who had also written a previous position paper. And I think that the conclusions of our study, even though this was 10 years ago, are very relevant to the new questions coming up with gene drives. So I've highlighted in red here what we're most concerned about are cases where there's little prior experience with the organism and the trait, and if the GEO could proliferate and persist without human intervention. Those are some cases that we need to be really careful about. Here's some examples of fish from a recent paper by Rob Devlin's group, showing you transgenic fish. These have not been released yet, but they're all the same age, and the small one is the wild type, and the larger one is the one with a growth hormone that's been upregulated. And these are examples of organisms that can get out into the environment, and it's tricky to try to predict what the consequences of this may be. The types of risks that we would like to avoid are listed here: creating new pests or pathogens that are more vigorous, harm to non-target species of any type, including soil organisms, non-pest species, birds, other animals, and disruptive effects on biotic communities, as well as irreparable loss of species diversity or genetic diversity within species. So these might be some ecological considerations with gene drives as well. With fish, I don't want you to look at this entire diagram, but this shows you how complicated the ecological questions are. If you start out with a genetically engineered fish, as the Devlin group and others have done, and try to anticipate what are all the effects of a new trait on this fish if it were to escape from the pens where it's grown for aquaculture into natural situations. And so they looked at a whole lot of ecological and evolutionary questions, ending up with trying to summarize these and what is the level of uncertainty, even after all this work in mesocosms and labs, and coming up with a recommendation or information that policymakers can use about whether to approve these products or not. So the ecological and evolutionary questions can be really difficult, and there's a lot of uncertainty involved in coming up with answers. These fish have still not been released after all these years of study, and there's many reasons for that. Okay, well, when we wrote the paper 10 years ago, I'm glad we put this sentence in there, because we said today's GEOs will soon be viewed as comparatively primitive. And that's probably true today, too, with what we're hearing about with gene drives. I mean, the field is moving so quickly. But this is a diagram of a transgene, the old-fashioned kind of transgene, and these types of constructs were used so widely in crops that the yellow region, the promoter, could be used actually as a label that you could look for with PCR to find out whether you had transgenic maize or transgenic rice. So very simple systems that we have experience with so far. And now we have CRISPR, so just a few illustrations about CRISPR, the focus of this committee. And obviously CRISPR is having a major impact on research with a very large number of publications, and you just see new applications all the time and new developments on how sophisticated the technique might be. I think we're going to see a lot of applications in agriculture of CRISPR for genome editing, but not necessarily gene drives. You don't need a gene drive to put new transgenes into agricultural crops. So what I'm going to focus on more today is gene drives for altering wild populations, and especially plants. And I think most people are aware that the Esvelt paper with this diagram of applications in mosquitoes is kind of the basis for these broader, more speculative applications of gene drives in wild populations. So suddenly, with this technology, humans have the ability to change not just the genome of a lot of different species, but also the balance of these species in communities, whether we want to suppress them or encourage them. So just to sort of get your head around that, I have these two illustrations. One of them is a tree of life with a person on the top who looks a little bit like George Church, but is really Charles Darwin. I thought that was interesting. But to put it in perspective, this is a tree of life phylogeny of all living organisms, and you can see that we're just a small part of this biodiversity. So now a quote from Esvelt is that we can do ecological engineering. I had never heard the term ecological engineering used in this way before I read this paper. It's something that has kind of been created to describe what might be possible in the future. In the past, ecologists always talked about ecological engineering in terms of creating restored wetlands and those kinds of applications. But now we've got genetic applications. So I'm going to focus on these new tools for ecology that Esfeld et al. Summarized, and many of you have seen the figure from their paper, which really helps to look at all the different applications we might be thinking about in the future. Aiding threatened species, controlling invasives, and agricultural applications like maybe restoring susceptibility to herbicides in weeds or making weeds susceptible to new chemicals. I was happy to see because of the cloud that I showed you in the beginning and my concerns about manipulating nature too quickly without enough knowledge, this paper and others have all had some kind of caveat in there about we need to be careful because we can't predict all of the ecological and human consequences of using this technology. But I think we have to make this even a higher priority as we go forward. So I like this next paper that is a response to Esfeld et al. in PNAS. It's an opinion paper. And this is by some researchers in Australia who work on classical species control with species that can control invasive species, so basically by eating them or introducing a disease. And these authors are asking whether CRISPR, instead of being a silver bullet as it's sometimes presented, might be a global conservation threat. And they are concluding that for the first time, humans, if we get good enough at suppressing populations, might be able to remove a species permanently from the planet. So that is something we obviously want to avoid, and I don't know if it's possible yet. I'm sure the committee will be discussing these issues. But I highly recommend this paper for outlining some of the scientific questions that ecologists have. I find myself needing to review some of the basic principles that you've heard about before, how a self-sustaining suppression that might lead to extinction would work. It's so counterintuitive to most of us when we think about natural selection. So the basic steps are outlined here of having a drive and having it inherited by all the progeny. This is for a self-sustaining long-term suppression of a population. And then whether that would actually work would depend on some of these characteristics of the system, the proportion of introduced individuals that have that, the number of locations where the drive is introduced, the generation time. If it's a very short generation time, then the drive might really be able to suppress the populations quickly. Effects of the gene that is being introduced on survival, growth, and fecundity. And I think some of the types of genes would be the skewed sex ratios and mate limitations, sterility. Many of these haven't been developed yet for most invasive species, but we can imagine that they could be. And some of the ecological questions down here are ones that I want to focus on, of what happens when you get outside the lab, outside the mesocosm, and you're talking about real-world environments. Do we really understand where these transgenes, where these constructs are going to go and what impact they're going to have on free-living, unmanaged populations? So getting back to plants, another fanciful diagram showing the diversity of plants. I'm just going to give you a very few examples because of the time limitations. But some considerations for plants. If a gene drive were designed to suppress populations of, say, agricultural weeds, it would work best in plants that have short generation times, like annuals or biennials. And here are a few examples of weeds. Horseweed, wild radish, and pigweed are all really serious agricultural pests that are annuals. Also, gene drives, of course, require sexual reproduction, and Fred Gould mentioned some of these terms in his webinar, which is a really good introduction. So if there's no outcrossing, if the species is mostly self-pollinating, like horseweed, it's not going to work very well. It's not going to spread the gene among individuals. But many species, like this wild radish, are self-incompatible, so they have to outcross. And this is a diagram of a pollen grain germinating and fertilizing an ovule inside of a flower. If the pollen grain has an allele that's different from the parent, then it can get through. And others have male or female. Okay, so there's a lot of different types of breeding systems out there, and it's these outcrossing ones that are going to be most effective. Okay, another consideration for plants is long-distance dispersal. Many plants do have very long-distance dispersal, and this would allow the gene drive to spread among populations. It's common in weeds and invasive species that wind and humans and animals are dispersing the seeds. And just a few examples, horseweed has wind dispersal, very long distance, up to 500 kilometers. Some plants like this tumbleweed break off and blow in the wind. And pigweed has been spread into that purple zone just by people primarily carrying this weed into a new area. Another consideration is whether the populations are connected enough for the gene drive to spread. So these are different populations that share alleles, but not this one, or these are completely isolated. And we can learn a lot about existing populations using DNA markers to find out how interconnected they are. And here's an example with horseweed, where the different colors are populations that are genetically distinct based on DNA markers. And this reflects some local dispersal as well as a few events of longer distance dispersal. Finally, hybridization is a big factor in plants, and many plants can hybridize with other species. These are just a few examples here. In this case, the native range of tamarix is shown here in different colors, but when it invades the United States, there's a lot of hybrids in the invading population. So we'll need to think about that when gene drives are introduced. So I think I have a little too much material. I want to emphasize that some of the claims of these early papers need to be carefully considered. And I don't think it's going to be very practical to introduce genes with susceptibility to herbicides into weeds. And just very quickly, there's a lot of types of herbicide resistance, a lot of mechanisms within a species. Glyphosate resistance can evolve extremely quickly over and over, so even if you were to repair the mutation, I think it would pop up again. And horseweed, which is mentioned in the Esfelt paper, is self-pollinating. So a lot of reasons why these techniques might not work. A bigger question that I'm concerned about is what if the safeguards don't work? And Austin Burt mentioned that. And I think that they assume there's a lot of connectivity between populations. The first drive might go to these yellow populations. A second drive may or may not reach all of the yellow populations, and then there could be a long-distance dispersal event that would carry the gene drive even further. So I think it's important to look at the amount of dispersal that can happen after a gene drive is introduced. Okay, just to look at then the big picture, these are some of the considerations that we need to consider is whether there's a huge need for the gene drive or if it's just a preference or maybe a way to make money. Would the effects of the gene drive be to suppress the population, or eradicate it, or could we even get extinction? Persistence of the gene drive effects in target populations. Would it just be for a few months in some of those self-limiting systems, or could you get longer, even centuries of gene drives in long-lived populations persisting? And is the distribution going to be very small and isolated, or could it be global? I think the examples on the left are the ones that should be considered first, and many of the others are going to be more challenging, and people who are looking at this from a regulatory perspective are going and doing the research are going to have a hard time figuring out when is it safe enough to introduce a gene drive into a wild population, especially for suppression, but also to enhance the population. I have some suggested reading, and these are my key take-home points. Simply to reiterate that these techniques are moving along very, very rapidly, and they're going to alter the population genetics and ecology of wild species in ways that we have no experience with, and you can't always predict from a model or a lab experiment what's going to happen out in nature. A lot of ways that they might affect populations, including enhanced fitness, of desired species. I think we need to look really carefully at the full range of applications, because some of them make a lot of sense, but others don't, and people all over the world will have different ideas about what is a reasonable application for this technology, and how could it be misused in some way that is going to cause major problems, economic, humanitarian, or environmental problems. I think there's a lot of potential for ignorance and human error, and maybe even intent to cause harm for these gene drives to be misused. So I'm very glad that the committee is discussing these big issues, and that we're looking into the future. We don't really know how many of these predictions are going to come true, or anything about the gene drive's effectiveness yet in wild populations, but I think these are really monumental issues, and I'm glad there's a lot of discussion about it, because I think they're as important as the human applications. Thank you very much.